बोलो मैडम ने बोला ये बात
Oh, nice picture. Phone is coming. ஆலமரம் ஒரு ஓட்டையில இருந்து எடுத்துருக்காங்க.
Before we move towards the next session, let me just reiterate our dilution, the final one.
Final destination.
So we had our sample library at 13.5 and our control library at 20. Right? Final required concentration was 9 picomolar. So when we say 9 picomolar, it is irrespective of volume itself because it's molarity.
Why we have chosen 9, sir?
Because for 9 picomolar, IBM is going to get around 900 to 1000 clusters.
Okay.
It will come based on your experience with the same instrument.
So, roughly we can assume that for 100 picomolar, 1 picomolar, 100 clusters.
Cluster density. That's the rough estimate. So what we do first is calculate the dilution factor. For example, if we had 10 picomolar to dilute up to 5 picomolar, so our dilution factor in that case is 2. That is, we are diluting it 2 times. Similarly, we have 50 and we want to dilute till 5. So we say it as 10 times dilution. So that 10 times, that is your dilution factor. So here what we will do is 13.5, that is our initial, divide by that is our required. This will give me 1.5. Similarly here 20 divided by 9. This will give me something around 2.2. Right? Now whenever we say we are diluting 10 times, that means part our stock and 9 part buffer. When we say 2 times dilution, that means 1 part of stock and 1 part. Similarly, when I say 1.5 times, this means that my library will be 1 part and my 0.5. This can be taken in terms of microliter, ml, liter, whatever unit you feel. Right? So finally, I am saying that my 1 microliter library plus 0.5 microliter. Therefore, 100 microliter library and 50 microliter of buffer. Up till you reach minimum value of 650. So I can have 400 microliter of library, microliter of buffer. That will give me 600. If I wanted exactly 650, similarly scale it up. 500 microliter 250 microliter. Plus 250. I am fulfilling all my conditions. I am getting exact numbers without the minimum volume is also 650, more than 650, and final concentration is 9 picomolar. That means that one part will be library and 1.22 will be my buffer. This because one part you have taken already of your stock library. So 1 microliter plus 1.22 microliter, scale it up, 100 microliter library plus 122 microliter of buffer. Again scale it up, 400 microliter of library and 488 microliter of buffer. Fulfilling all the conditions?
880.
Doesn't mean. Minimum 650, final volume 9 picomolar, and exact without any decimal. And finally, I had 750 microliter of my library, sample library. I am going to add 2 microliter of my control library. So I took 15 microliter, removed 15 microliter of this, added 15 microliter of my control library. Quite simple. Not doing right. Anything to ask first? Yeah.
समझ में आया कि बात हो
That is clear or
clear plus.
okay. So we'll move on towards our next session, which is on the biological file formats. Since you all are going to work on hands-on data, this session is one crucial building block for your data analysis. Now, most of you are not aware and don't have the computers and all. So understanding this file format is a necessary thing, right? And Dr. Ketan will these file formats, how they are generated, what they represent, how the representation means, how you can understand them, and all the other details.
Yes, it's function. It's a function. Charlie? No.
Laughing, they know Punjabi.
Everybody else might have seen Mahabharata.
So you know the IST.
Sir, you have specs. He's not there.
वो मुझे बाहर दूंगा दिखता है तो मालूम करता है अभी
And looking about this.
Sorry.
So, files are the soft copy which stores our data. Depending on the extension which you are using, it gives you an idea which information it stores. If you know that we are using for matrix analysis and genetic sequencing, that what we have is genetic material of library, sequencing library, we do the sequencing run, right? After sequencing run, what we are getting is, and from with the help of raw data and different bioinformatics pipeline, we do data analysis, and the data analysis gives us something that is of interpretable, and that interpretable will give us the finding what we have looked for. exemplify that printed paper. Printed paper is a material in which the information, but although they are printed, they are categorized in different forms according to the information they have. The left top is the magazine, then newspapers, then our textbooks and novels. Though all four are same material, but the information they carry on the basis of that they are differentiated from among. Right? So by looking at it, one can find that I'm not going to get any informative news from this, and vice versa applies for. So that is the case, and it comes to us that why we need different formats. Different types of software do frame or do conceptualize in a way to handle different types of data. Now, all the data cannot be fit in a single format. So, there is no format because the file and the information varies from your extension to extension. Second, representation of data. You can interpret your data with the help of bar graph. Very well. But if you look at the same data in form of text or numericals, you won't be able. That same applies over here, that it gives you a better presentation of your data. The third is conversion of file format access and sharing. I mean easier access and sharing. Let me give you an example of opening having got MP4 or MK extension of size of 4 gigabits. That is 4 gigabits. It will take time. But instead, if you open 250 KB of .wav, or I would say .g, would take less time. Why? Both are video file on the basis of their size as well as their readability to the system. So it is, if the information is easily accessed to the software which is opening, then it will open very soon. Other time. But again, it will depend on your size and the data it has. Right? It supplies required information for each step of analysis. What I does mean of that is that on the basis of extension, I am particular with the bioinformatics analysis, that on the basis of the extension, you will come to know that which stage you are at in the pipeline of your analysis. Meaning that if I give you two files, one file is having extension of FastQ, and second file is having extension VCF. By looking at the extension, you can know that FastQ is our raw file and our VCF is our final file. Because we do know that what the FastQ file contains, what VCF file contains. So for that, one should have an in-depth knowledge. I'm not saying that you must know in which line it tells which means, but I'm saying that you must have an idea of the overall structure of each file type, which are major this analysis. So on the basis of the type, we are categorized into 5 file formats. That is row coordinates file, parameter files, annotation files, and mechanical files. You all know that what we have generated from instrument next generation sequencing machine, that is our row sequencing. Coordinates file. It is the file which we use generally for mapping purpose. Right? Third one is parameter. In upcoming session, we will come to know that different parameters are given to the software which you are using. So in software, they are required they will ask you to define your parameters in a file. file you need to provide to the software in order to run your data. So that type of file will be used for parameter files. Is annotation file. The annotation file will have exact information that this particular gene or protein kind of sequences, right? And the last one, metadata. In the previous day, Subhas sir has overviewed that what is the metadata, because the variable, as much as variable you are having for your samples, of doing multivariate analysis will be more, and the analysis, interpretation from that analysis would be more informative to the research community, right? And the last is each data formats vary in the information they contain. As I said, .fastq and .vcf. So, on the basis of category different file formats, the highlighted one are the formats which are majorly used in biological file. That is FASTA, WALL file, FASTQ file, SAM file, BEM file, DCF, Delimited. And these four, GFF, GTF, MAF, and BED, they are the files, but they are intermediate files. No one bothers to see them or to pinpoint the errors they are getting during the pipeline analysis, right? So, before that, we are having a knowledge of phrase program. So, the base calling accuracy of S is based on the phrase code, right? So on the basis of phrase code, one can have idea whether your sequence quality is good or not. So for representation, this is an equation which gives you the probability of error sequencing. Meaning to say, if your phrase code is 10, it does mean there is a probability of error of one base in 10. If the phrase score is 10, that does mean it has a probability of 90. And likewise follows for 40. That is, a error possibility of error of one basis is having accuracy of 99.99. Right? The concept which you should be introduced is the ASCII coding. So the introduction says that there is a standard code in which numbers stands for alphabet, special character, and the digit. If you carefully look at the background image, you can have an idea that different ASCII code have been allotted to different kind of symbolic character. Say for example, for 40, it has the ANSI, that is stands for American National Standard Institute, represented this character set, and starting from 1, this is 1 to 31 are the control characters. From 32 to 1 are the special character that contains uppercase, lowercase, and other symbols. So this is the range which are used most of the next generation sequencing in order to justify or represent that quality score. And from 128 to other symbols. So in order to explain this, pay attention. So this S is for the Sanger, right? The green one is for Solexa, I for Illumina, and again orange is for also Illumina, and the red one is for Illumina. This is the software version or the version they are having. So if you say for Sanger, it goes from 33 to 0 to 40, and likewise for. In this case, if the I, J, and L, all three are from the Illumina. So if you have done your sequencing in 1.3 plus, right? And if you want to convert your RC into 1.5 plus, then we are having a difference in the encoding system. So they are very possible in order to convert your ASCII encoding from 1.3 plus to 1.5 plus, right? So with that ASCII encoding and phase code, let us have a look at the first file format, that is FASTA. So it is a text file format representing nucleotide or the protein sequence, right? Nucleotide is ATGC and other for the combination of different nucleotide, and the protein sequencing from different amino acids. We have there is .fa, .fasta, .fna, .faa, and .fsa. If you open any of your text editing software in your laptop or PC, then you will have a structure like this. That the first line has greater than signs, followed by an identifier name of a sequence, that is header line, and it has a nucleotide sequence or amino acid sequence. In this case, we are having nucleotide sequence in this second line. If you repeat this structure after one another, then this is the multiple file in file. So if your slide is one sequence, then you will have multiple lines of your sequence. If one, then it is one sequence enough sequence. But in a later on, I will get to back to you that how you can manipulate the sequence and that is greater than sign, the identifier name and the sequence have importance while doing your analysis. And I will exemplify you that we will have our last 10 example. Second one we are having is master file containing the raw sequences contain a quality score of each basis in the corresponding cluster. So in previous we are having master file starting with greater than sign. The same goes for the whole file or nucleotide sequence. It is having a corresponding quality score of that particular nucleotide at particular position. If you are having, say, 8 nucleotides in the field, then we are having 8 numbers represent this code. And this must not vary. Otherwise, if you want to compile your FASTA file into FASTQ file, you will get an error.
FASTQ file is combination of FASTA and FASTA.
Yes. That was the file which we were getting before in the first generation of next sequencer, but the sequence using right now are giving us file in FASTQ format only. So in the third one, the FASTQ is instituted for storing sequence as well as for quality score. It has extension that is FASTQ and FQ. Now, a single nucleotide sequence comprising of 4 lines. Line 1 starts with @sign, which is a sequence and with additional description. The second line is raw sequence. The raw sequence of this code, which is in the fourth line, are separated by the cluster, right? So in your file, if you are having this, if you divide them by 4, you will come to know that you are in that file you are having 25 sequences, right? Mark as the identifier of your sequence. The long name which you are seeing over here, it is MiSeq, and each information between this column is of different position, different cell, and there's X, Y, Y, and Z axis, right? And corresponding to key, we are having different ASCII encoded value, right? If you see accurate sign, the accurate sign is for, can you look at from the background image?
There is 64.
Yes. Third line, first, 64. So, accurate is for 64. But we could not have 64 as our polygon. But if you refer this, then you can make out that according if it is 1.3, then 64 will be 31, our quality score. We are getting a point. We are having two file formats which are durable system, like durable system. The first one is SAM, DAG. The SAM is sequence alignment, right? And this another one is binary sequence alignment. Now, what are we identifying your variants? If you look, then we are having sequence read instrument in form of FASTQ file. When you do a mapping, reference-based mapping, then after quality control, you are getting this .SAM file and .PIM. This file are used again for variant calling and it will give you. So on the basis of the extension, as I mentioned before, you can have an idea that at which stage you are on the. If I am saying that I am having SAM format and time format, then you have an idea, okay, that person has already done the alignment part. I am saying that I have this here file, that one can have an idea that I have got the variant calling analysis, right? Now, the reason is why because few of these steps require high computation power, right? Editing your FASTQ file, quality control your FASTQ file, and extending variant calling won't ask you more computational power. But if you are going for alignment of your genome, and then it will take you more computational power, right? So in that way, you can manage that which step is performed at the computational system. The SAM is also a text-based format for storing your biological sequence, which is aligned with the reference sequence. I will get to the structure of that one file, SAM file, and the BAM is the binary version of your SAM file. So if you open the SAM, we can read it. But if you forcefully the BAM file, you cannot read it in the text box. Right? Extension, as I mentioned, it is .SAM and .BAM. Right? The structure of both the files are divided in the header part and the alignment part. So, this is the reference, and these are our 4 reads, which are aligning at the reference at 4 different positions. This is a representative idea of our alignment part. That over here we are having the header part. The first that is starts with the @ sign would be our header part, and the part which starts with our read name will be our alignment part. Now, if you see the header part, very simple. That is header line denotes different symbol. I have exercise for the presented one that @SD stands for header line, @SQ stands for reference sequence, and likewise for this over here, and this over here. That is SO for sorting order and for reference length. Are there, but it depends on the number of reads you are utilizing and which reference you are using, right? In the alignment part, as I mentioned, this is structure, the error which I have seen, and the alignment.